Okay. All right, and then share the screen. All right. Okay, so my name is Zhen Yujia. You can call me Arthur if that is easier for you. And Lei is the TA, and he's a PhD student in my lab. So we are doing research together, and we are also doing teaching together. So this quarter, we're gonna be here to help you explore genetics. All right, so this is our textbook. I hope everyone has the textbook. And we also recommend the solution book. And this solution book the manual book can help you do the side problem, which can help you better understand the topics covered in this lecture. And so the lecture time is every Tuesday and Thursday in the morning, 9:30 to 10:50. And yeah, this, of course, you already knew this because you have successfully logged in to the Zoom meeting, right? All right, so let me show you something. Oops. Let me share the syllabus here. Okay, so this is the course syllabus. I have already uploaded this syllabus to the iLearn website. So if you log in, you can check it out. And this syllabus gives you the basic information about this course. And the most useful information is on the second page. So so for each lecture, we listed the topics to be covered in the lecture, and also on the third column, the fourth column of this table, you're going to see the page numbers here. So those page numbers are the side reading assignment. So that's my suggestion. So if you, it would be better if you can read those pages before you come to the lecture, because this will get you well prepared for the lecture. And the topic, the topic is to be covered. And also, for each lecture, I put in some problems. So the questions on the back of each chapter. So for example, chapter 2, so there's a solve the problems. So this solve the problems means there's not only the questions, but also the book shows you how to solve this problem step by step. So it's very, very super helpful. And there's some other problems, but you can find the solutions in the manual book. And yeah, I highly recommend that you read textbook and practice those problems, because this will help you understand the topics covered in the lecture, and also can help you well prepared for the quiz and the exams and the final exams, midterms. And so this is the book, and please read this book. I think like reading the book is not only for you to get a good grade for this class, but also for myself, I found it is fun to read this book. This is a very, very good book. It's well written and very clear. So I hope you enjoy reading it and benefit from reading it, not only for the class grade, but also for your interest in the genetics world. All right, so let me check. Okay, the grade breakdown. Yes, of course. So some student asked the question about the breakdown. So so on the first page, as you can see, on the first page of the syllabus, this is the breakdown, the percentage of the letter grade assignment, just like this. And so this is just a guideline. So at the end of this quarter, so the letter grade assignment will be determined at that time based on your points. Okay, so this is how we break down your grade here. So the total points, the maximum possible points for each of you is 300 points. And this includes 75 points from the discussion session. I'm going to talk about this a little bit later. And the midterm, we have 2 midterms, 1 and 2. So each one is worthy of 75 points. And then the final, the same, 75 points. Let me go back to the slides. So I'm going to show you something about the discussion. All right, so will the test, another question, will the test be on materials in the book, and your lecture slides? Yes, of course. So because there are too much stuff in the book, so I don't think I can test everything in the book. And as you can see, based on the syllabus, I don't cover each individual topics in the book. Because this is, so to teach the whole book, so this is not a quarter, just a 1/4 thing. So focus on the topics I selected for you, which are listed on the syllabus. And I'm gonna test you based on the lectures and the the reading assignment, and also the side problems. So, because yeah, sometimes like on the test, on the quiz, or on the exams, you may see some questions which are very similar to the side problems on the book. So that's why my suggestion is, yeah, please do that. It'll be fun, yeah, and also it will help you to get well prepared for the exams. Okay, let me see. So I am not. So let me see. I hope I didn't miss any questions. So maybe I don't have time to answer each of your questions. So, but if you like, because I try to keep the pace of the lecture. But if I didn't answer your question in the chat box, if I missed some, so please, yeah, send me email afterwards. So I will try to answer your question later. Because usually Lei and I are working very hard on our own research during the daytime. So we try to allocate a significant amount of time during the night to respond to your emails and to answer your questions. All right. Go back to the slides. All right. Okay, so lectures. So for the lectures, I mainly focus on the topics I selected for you. So this is the center of the class. Make sure you understand all the listed topics for this class, and take notes, please. All right. So this will be very helpful. All right. Discussion includes the quizzes, and also TA will help you to review the topics that we learn in the lecture, and also can help. He can help you with problem solving. For example, the side problems. If you have questions, yeah. So this is the time and the place that Lei can help you to do those problems. And so for the first week, tomorrow, I believe each of you has enrolled in one discussion session. So yeah, please make sure to attend tomorrow's discussion. Although there's no quiz tomorrow, but your attendance is important because your attendance will be used to confirm your enrollment, and this is worthy of free 5 points. So that's the easy points to take. Don't miss it tomorrow. And so usually we have a so-called genetic clinic for everyone. If you have questions, this genetic clinic will be held right before midterms. Any questions? Okay. And so we're going to very actively use this course website. iLearn is, as you can see, so each time if I make an announcement, if I put something new for the class, I'm gonna use this website. So please frequently check this website to make sure you get all the learning materials. Okay, so we have covered this part. So this is how we break down the grading system, right? Okay, so discussion format. Usually discussion will include a short quiz, about 15 minutes. And this will be a small test based on the previous lectures. So you can check the syllabus to see the schedule and the topics to be covered in each quiz. Again, tomorrow, yeah, there's no quiz for week 1, but your attendance is required because yeah, this will make you to easily get a free 5 points. Yeah, don't miss it. And the quiz, so TA will decide whether a quiz is given at the start or at the end of the discussion. So this is up to Lei. And for discussion, the main activity will be the problem solving led by a TA. So sometimes they can for each section, so discussion session, they can divide students into groups of 5. So each group will be assigned a problem, and you can work together to solve the problem, and you can pick one representative to answer the question. So in the discussion session. So this is how we organize the discussion in the past and also for this quarter. All right. So I believe that this is a important question to everybody, right? Because you want to take this class and you also want to be successful in this class. So those are the tips that you can use to improve your performance in this class. Of course, you need to read and understand the syllabus and also for the class procedure. So those are very important. Each time I will upload the lecture notes, the PowerPoint files to iLearn before the class. Yeah, you can print those lecture notes and read it beforehand. And so during the lecture, you can write the notes on the lecture, write your own notes on the PowerPoint file, right? And this will be a very useful material for your review to prepare for the quiz and exams. So come to the class. So this is very important, and get the easy points. For example, tomorrow. So tomorrow, no quiz, but attendance can help you get 5 points for free. And also the quiz points are very easy to get. So don't miss those. Those are easy points as well. And it takes up about like 25% of the total. So yeah, very important. And textbook. So reading this textbook is fun. So I enjoy reading this book. So it's really very well written, and so hopefully you also enjoy reading this textbook. And do the assignment. So we do not have, technically we do not have this. For this class, oops, we do not have assignment, but I do give you the side problems, so it's up to you to do this. But I highly recommend that you do this aside problems, because this can help you better understand the topics. And sometimes you're going to see the questions shown on the exam will be very similar to the side problem. So yeah. So this is my suggestion, and also try to link the knowledge principles you learned from the lecture to the real problems, which means you need to understand the principles and know how to apply these principles, your knowledge learned from the lecture, to solve those problems. And of course, so you will have questions, you will have a difficult time to understand the topics or to solve the problems. So bring your questions or problems to our office hours or genetic clinics. So we are here to help you to solve the problem. And so yeah, you don't have to fight alone. So you can study with a group. Yeah, but the collaboration is not allowed during the quiz and the exams. So other than that, so yeah, you are encouraged to study topics in group, because support from other students is very useful. And finally, to better understand the topics learned from the lecture, it's very good for you to discover genetics in the news and around yourself. All right, so those are the tips that I can give you to be successful in this class. All right, so any questions? Let me see. Oh, wow, lots of questions. Okay, thank you. I don't know, like exams, probably, yeah. So this is challenging for me, because this is my first time, but so we're gonna figure it out. So Lei actually taught the same class last quarter, so he has experience in the exams. So I'm gonna talk to him, and we will. So it's very early to talk. Still early to talk about this topic, right? The exam, but yes. So we're gonna figure out a way to do the exams, and all right. So okay. All right. So about about the quiz. So probably because tomorrow there won't be a quiz. So I think Lei can spend some time to discuss with you how he can organize the quiz for you in a nice way. So all right. So I'm gonna leave this question to him. So you're gonna talk to him about this about the quiz. All right. Let's continue. So lecture one. So today I'm going to give you a very brief introduction, and we're going to also cover the single gene inheritance, and also the mitosis and the meiosis. All right. So this is a genetics class, and what is genetics and what is its history? All right, so actually the genetics, the GEN is from Greek. It means to become or grow into, and this this discipline or this field is actually the study of the, so genetics is actually a study of genes and the genomes. So it is also called the science of heredity. Okay, so this is the history of genetics. Of course, when we talk about genetics, we must, so this is the must talk person, right? Mendel, all right? Because this gentleman actually set the fundamental basis for the modern genetics by doing a series of experiments on the pea plants. All right, so as you can see here, he did a lot of interesting and also very important experiment based on pea plants. He checked a variety of the phenotypes for different sections of the pea plants, and he studied the important phenotypes here. So, for example, we're going to talk about this later. So, he studied in the color of the flowers, purple versus white, and he also studied the shape of the seeds, for example, the round shape or the wrinkled shape, and also the color of the seeds, yellow versus green. And all right, so he proposed a hypothesis like this phenotypes can be inherited through something that acts like a particles. And right now we know this particles is called the genes, all right? So this is his very famous pea plant experiments. And okay, so now after that, so he just proposed something called the particles or genes that carries the genetic information, and it can be passed from generations to generations. But where is this particles located? So the second gentleman, the second important scientist in genetics history is Morgan. All right, this gentleman actually did a very creative experiment to figure out that the genes or the Mendelian particles are actually located on chromosomes. So his contribution is to identify the location of the genes, which is on the chromosome. And after that, Fisher actually, so is a statistical geneticists. So his contribution is to try to use Mendelian genes theorem actually to expand the Mendelian gene single gene theorem to the multiple gene theorem to explain a complex traits. So for example, here, Mendel actually focused on one gene, one phenotype, but Fisher expanded the theorem to multiple genes to a complex traits. All right, and after Fisher, there's a theorem called the one gene, one enzyme. So genes, a specific gene can encode an enzyme, and this enzyme can function and have a very specific function in cell. And however, those genes can be involved in a pathway to affect a single trait. So this is what we're going to learn in the future. All right. So till now we know, okay, gene is the particle that carries the genetic information, right? And this genes can be passed from generation to generation. So what does gene look like? And how the genes can be stored in a cell, in each of the single cells. All right, so in 1950s, I believe, oops. All right, the Watson and the Crick, they figure out the structure of the genes. And so they figure out the structure of the DNAs. All right, and we're going to talk about this a little bit later. And so this is the history of genetics. And all these important contributions lead to the current modern genetics, different genetic theorems and also genetic applications. For example, those are the terms which are very probably you are very familiar with. So the GMO, the genetically modified organism, and also the sequencing technology, right? So, for example, for human genomes, we can use the current advanced sequencing technology to get the sequence for each individual, and then we can check those information to find out the reasons for diseases or disorders. So this is a very important and hot applications currently. And also, yeah, this is related to the medical genomics and then personalized medicine. And also we can, right now we can use advanced technology to fulfill the gene therapy. And also we can change the genome by genome editing. All right. So this is the history of genetics. All right. So by studying genetics, we need some tools. Those are the commonly used tools for helping us to understand genetics or to help us do genetic analysis. The first one is the in vitro experiment, which means in a lab, so we can leverage the cell's own machinery to do like the DNA copying, the pasting, or cutting, something like transcribing DNA. So this is one way we can use to help us to do the genetic analysis. And also we can do the clone DNA and transformation. So transformation means like, so we can insert some foreign DNA from one species to another species to change the features of the target organism. So this is called the GMO. And also we can use another tool we can use for genetic analysis is through the hybridization. As you know, so the DNA can bind to DNA. The DNA also can bind some other molecules like the RNA. So we can leverage this kind of features to do experiment and also to understand the genetics. Sequencing, yeah, this is so hot, so popular. So I don't want to, so go deeper, but yeah, most of you have already heard of this, right? Sequencing technologies. So if you put sequencing in the Google search box, so you're gonna end up with tons of information about this technology, and also genomics. So genomics is a study or methods for analyzing the entire genomes for an organism. And this genomics involves, so the genomics actually is an interdisciplinary field. It involves a lot of other fields, like for example, the bioinformatics, computer science, and also statistics. So we're gonna talk about this in the future. So now today, so what genetics can help us to help people? So first, we can use genetics tools to see how organism or how human evolves, right? And also we can learn why people in different region are affected differently affected by the lactose intolerance. And also we can learn why and how people can be adapted to the life at a very high elevation. So this is what we can do to use genetics to study ourselves. And also we can use genetics tools to help us to study a human disease, trace a disease. We can use genetics to help us to trace a disease gene through a family tree. So this is a very important but very difficult topic. So we can talk about this in the future. And also we can use genetics to improve, for example, food for human beings. This is so important for the current world now. Okay. All right, so besides this class, so our class is Biology 102. So besides this class, we have a lot of genetics classes on this campus. So if you are interested in this topic, genetics, so in the future, you can choose different classes to explore more about genetics. So this class and also another class. Okay, so yeah, I'm gonna try to slow down a little bit. Sure, thank you. All right, so this class is mainly focused on the transmission, which means we will study how the genetic factors is passed down from generation to generation. So there is another class. So if you are interested, so probably in the future, you can consider taking that class as well. That class is focused on the chromosome genetics. And also some other classes like the Biology 107. So it is focused on the molecular genetics or the mechanism at the molecular level, which can help you understand how the genetic factors are coded and how these genes are expressed at the molecular level. So that's the molecular genetics. And also you can find the evolutionary genetics. So this is more focused on the high level, the population, individual and the population. So in this class, you're gonna see the change of genetic factors over time through the mutation or recombination, and these mutations, how these mutations are under the selection stress, and yeah, the whole the evolution history for the entire population, something like that. And also we have class to teach human genetics. So we're gonna see how the basic genetic principles are applied to ourselves, to human beings. And also we have some courses which are focused on the genomics and bioinformatics. So in this class, you're gonna see how we can leverage the genome sequencing technology. We get the humongous data, big data, and we use some advanced technology methodology to analyze those big data to help us understand the genetics behind the phenotypes, variability, or the human disease. So help us to understand the genetics behind human disease. This is another class. So sometimes I teach this class as well, the quantitative genetics. So this is a class to study the statistical genomics. And also in this class, you're gonna study, be able to study the polygenic traits, the traits that are controlled by not just a single gene. So in a minute, we're gonna start Mendelian's study, right? That's a simple case. That's a simple trait, which is controlled by only a single gene. But in this class, we're gonna study the complex traits, which is controlled by more than one gene, more than one loci. All right, so I think I just finished the introduction to show you what other opportunities for you to further explore genetics on this campus. All right. So I believe everyone has learned the very basic biology to know this topic, right? The genetic material is DNA. And so DNA is actually two strings. And these two strings are held together by the hydrogen bonds and form into a very nice double helix structure like this. And so each strand is actually composed of a series of nucleotides. And if we zoom in and look into each single nucleotide, we're going to see each nucleotide includes a sugar, the oxyribose, and also a phosphate, and one of four one of four bases, right? A, G, C, T. All right, and the nucleotide sequence of the bases in DNA molecules actually encodes the genetic information. So this is what we have learned before. All right, and so in this class we're gonna focus on so our focus for this class will be on the nuclear chromosomes of the eukaryotes. All right, so the DNA molecules can be packed into a very nice molecule or very specific structure called the chromosome. So we're going to see what chromosome is. So here, so as you can see, usually chromosomes exist in pairs in most of the diploid organisms, including ourselves, human. All right. So this is example for human. So in each of our own cell, we're going to see there are 22 pairs of chromosomes. Those 22 are called the autosomes. And we also have another specific pair of chromosomes here. We call this sex chromosome, right? And the chromosomes exist in pairs. So for example, like this pair, they have two chromosomes. And we call these two chromosomes as the homologous chromosomes. So one homologous chromosomes is from father, and the other one is from your mother. All right. So if you look at these two homologous chromosomes at each locus or at each position of this chromosome, there's a gene. There's one gene, and this gene may take different forms. So I use the same shape, the square, to represent the same gene. But here I use different color to represent the different forms of this gene. Different forms of this gene, we give these different forms a name called the alleles. So different forms of the same gene, we call this different. We call them different alleles. So for example, in this example, at this locus or at this position, the two alleles are identical for this gene, right? The same shape and the same color. But if we look at this locus, so the same shape means the same gene, right? A different color means different alleles. All right, the same thing here. Those are very important concepts. So sometimes in the lecture, when I say something is important, you better trust me, because this is very important. And I probably will give you some questions about these concepts in the quiz or in exams. So you need to understand the concepts of chromosomes, homologous chromosomes, and the locus. And at each locus, there's a gene, right? The gene can take different forms. And those different forms of a same gene are called alleles. Anything wrong here?
Professor, I had a question.
Yes.
So on this diagram, so there are two locuses and three kinds of alleles, or at least allele shapes.
So in this example, right? Actually, in this example, we have three loci. So this is the first loci. Let me label this one. This is the second locus. This is the third locus, which means we have three genes on this chromosome. But when you look at each of these locus, so for example, the locus 1, we only have one allele because the color is the same, right? But for the second locus, so now we're talking about a different gene at the different locus on the chromosome. But here you see, so the chromosomes, homologous chromosomes, both have the same gene, the same shape, but different color, means the alleles from father is different from the allele inherited from mother. Is that clear?
Yeah, I get it. So the alleles themselves are just the colors for each one of the shapes.
That's right. Yeah,
Okay.
very good. Yeah, thank you. The shape, each one of the shape represent a specific gene at a locus, but the color shows if the color shows if these two alleles are identical or not. Any other questions here?
Yeah, I just had one more question. Oh, I'm so
Mhm.
sorry. I'm just a little bit confused on that again. So the first one, if it's the same color and same shape, then it's one allele.
Yes.
But if it's a different color, then there are two, right?
Yes. Can you say that
Yeah,
again?
thank
Because
you so
your
much.
voice is kind of like broken because of the internet. Could you ask your question again?
Yeah, so I just wanted to clarify what you have gone over so far. So if it's the same color and same shape, then it's one allele, right? But if it's a different color, then there's two alleles. So the first.
That's right. Yes, correct.
Thank you so much.
Oh, sure. My pleasure. All right. So this
Professor?
is very important. Yeah. Any other questions?
So yeah, I just wanted to, so between homologous chromosomes, each chromosome or like in that pair will have the same genes at the same locuses, but you have the potential of having different alleles of those genes, right?
That's right. Yeah, exactly. Yes.
All right, thank you.
Sure. So again, this is very important. I hope every one of you understand this. And this is very important and can help you. Yeah, understanding of this can help you solve a lot of important genetics problems.
And for the sex chromosomes, since they don't have a pair, would you still have the same genes at the same locuses? But
Yeah, this is a very good question. So we're going to cover this later. Because
Okay.
because this is a very special pair. So we call this sex chromosome, right? It's not the same with the rest of the chromosomes, which are called the autosome. Because as you can see, let me clear this and clear the slides. Oops. Yeah, so if you look at any of the autosome pair, so they are of the same length, which means like at this locus, if there's a gene at this locus, you must find another, you must find the same gene at this locus on the other homologous chromosomes, right? Because they are similar. However, if you look at the sex chromosome, as you can see, the length of X chromosome is significantly larger than the length of Y chromosome, which means probably only part of the X chromosome has the same gene on Y chromosome, but some of the genes on the X chromosome does not have the same genes on the Y chromosome. So we will pour this, we will use a different term to name these genes. So we're going to cover this later, not today. All right, very good. This is a very good question. Any other questions about this? Very good. All right, so let me give you an example. So the example from Mendel's experiment. As I mentioned, so one of the traits that Mendel studied is the flower color of the pea plants, right? Right, so these are the two homologous chromosomes. And if we look at this locus, there's one gene. There's one gene which control the color of the flowers. And so why do we have like one gene shown white in one chromosome, but the other one is the pink or the purple on the other homologous chromosome? What does this mean? Can anyone help me? What does this mean? So the same gene, why different color here?
Means there are two variations of that gene.
Yes, two alleles, right? Very good. All right. So we can name this purple allele as the big A. So So we're gonna study this. Why we put big A here? Because we call this allele as a dominant allele. All right. And this white one, we use the small a. We call this recessive allele. All right. So if a individual plant, pea plant, carries carries big A and a big A on both homologous chromosomes, so of course the dominant allele will be expressed, showing the color on the flower, right? And the same for this heterozygous individual. So even this one has a recessive allele, but this is a recessive one. Yeah, on the same time, this individual also has a dominant allele. So when these two alleles come together, so the dominant allele will be expressed, and then the recessive allele will be hidden. So still, you're gonna see the purple color for this individual. Is that right? However, if an individual pea plant carries two little a alleles, two recessive alleles, then this is the only opportunity for this individual to express the recessive allele. So you're gonna see no pigment be deposited in this flower. So you're gonna see white. Does that make sense? Okay, by showing this example, I just want to help you understand a single gene at a single locus on the chromosome. However, this gene may take different forms, alleles, and there's a relationship. For example, here we just talk about a simple example, right? We only talk about two alleles, but in reality, at a single locus, the gene may have like more than two alleles. So there's a very complicated relationship between each allele pairs. Okay, so this is a simple example. I just show you a very simple relationship between these two alleles. One is the dominant allele, which is also called the wild type, because this allele represents a gene that is functional, that is functioning, and it can produce the purple pigment, and this purple pigment can be deposited in the flower. So that's why you see the purple flower. However, the second allele, which is the white one, so if you look at the DNA sequence of that allele, it looks very much like the wild type allele, but there's some mutation inside. But those mutations are very fatal. It can totally like destroy the function of this gene. And the allele like that, the mutation cannot produce pigment anymore. So that's why, so if an individual has that kind of mutant allele, so this individual cannot produce purple pigment. So you only see the white flowers. Does that make sense?
I just had to understand if we had the white flower with the two lowercase a and the purple flower with the capital A and lower a, if those two between those two, the purple flower would be the dominant, and if they were to be somehow crossed, the flower would be purple because of it.
So if you just cross these two, the big A, small a, with the small a, so yes. So if you generate a large population of the progeny, you're going to see half of the progeny will show the purple, and then the other half showing the white flowers. Does that make sense? We're going to talk about this. So your question is a little bit advanced. we're gonna talk about the cross experiment a little bit later. Good question.
Okay, thank you.
Sure. All right. What time is it now? Okay, let's move on. So I hope I can finish all the slides today. All right. So genotypes. All right. So the flower color is called the phenotypes. So something we can observe. And this phenotype is actually determined by the genotype. Genotypes means the specific genetic makeup. For example, here we use the big A to represent the genotype for this individual, right? So this genotype will give you this phenotype, and the same here. All right. So here, this slide, I just want to show you like the relationship between the genotype and the phenotype. The genotype will determine the phenotype, and the phenotypes may be inherited through the genotype. So as just I mentioned, in a simple example, there's some relationship between the two different alleles. This is the very simple relationship, right? The dominant. For example, the flower color. In human, some disorders like the dwarfism or the lactose tolerance, those are the dominant relationship between different alleles. And also the recessive. Recessive means like some phenotypes will be masked in some generation. For example, this is a human disease called the cystic fibrosis, which means like if we use A to represent the wild type and the small a, lowercase a represented the mutant, the recessive one. So the individual only, the individual with little a will show the disease phenotypes. So any of this individual will not show the phenotypes, disease phenotypes. So this is the dominance and the recessive. And in the future, we're going to also learn some phenotypes that are sex-linked, which means the genes causing this disease is located on the sex chromosomes. So such that the inheritance pattern or the expression of the disease will be influenced or will be determined by the gender. So this is the future topic. We're going to talk about this. All right. And sometimes, yeah, the harmful traits, alleles as well. Okay, so when we talk about the traits, the phenotypes, some traits could be very complicated, complex. So for example, yes, question? Okay. So for example, a single gene, a single genetic factor can cause more than one phenotypes. So we're gonna learn this disease, the PKU. So the gene, this gene, if something goes wrong within this gene, then it can cause more than one phenotypes. For example, it can cause the mental retardation. Also, it can cause the reduced hair, or it will affect the skin pigmentation. So we have a term for this kind of traits. So please, yeah, this is very important. Please pay attention to this term, the pleiotropic, which means a single gene can cause problems or variations in more than one trait. So this is called the pleiotropic. And another type of trait is is showing the genetic or locus heterogeneity. So, which means a single phenotype or genetic disorder may be caused by one of multiple number of alleles, or so when we talk about multiple number of alleles, here we are only talking about one gene, right? Oops. One locus. Yeah, which means there's a wild type allele, so which will not cause any trouble. But if there's there are multiple mutation forms for this allele, it will generate multiple alleles. and those alleles may cause trouble for this gene. Or there's another, oops. Okay. Sorry. Or this phenotype may be affected by the non-allele mutations, which means, so when we talk about the non-allele mutation, so here we are talking about more than one genes. So this is a chromosome. So there's one gene at this locus, there's another gene at this locus. So the disease may be caused by the mutation in 2 genes. So this is the genetic heterogeneity. And also in the future, we're going to talk about the quantitative traits, which means a trait is controlled by more than one genes. And we can use the genetic model, the linear regression model, to help us to understand the relationship between these traits and the different causing loci, different genes. And also, one thing I would like to point out is the environments, the importance of the environments, because the environment contributes to the traits as well. And in the future, you're going to learn the variability observed in phenotypes sometimes will be caused by the interaction between genotypes and the environments. Let me check questions. Thanks for the questions, but I think I'm a little bit behind my schedule, so I don't think I can answer all of your questions now. So yeah, please, after this lecture, if you still have questions, please send those questions to me or to Lei. So we will try our best to answer your question on time. Thank you. There's some problem with the slides. Let me stop sharing and do this again. Okay. So as I mentioned, we can use a linear model to study the relationship between the phenotype and the genotype and the environmental factors and their interactions. Right. So we can use this this linear model to explain the relationship between the phenotypes and all the factors. All right. So here. Here, Y is the phenotype, the G is the genetics. Phenotype, the E represents the environmental factor, and the G by E means the interaction between the genotype and the environments. And the epsilon here represents the random error. So random error means noise. Yeah, random error events, we also call this noise. Sometimes it can cause very small effect on the phenotypes when compared to the genotypes or the environmental factor. For example, so let's use this example to show the noise or random effects on the phenotype variability. So all cells in your face have the same genes, right? Identical genetic makeup. However, they probably, yeah, and also, yeah, all the cells are developing in a very similar homogeneous environment. But why your face do not look perfectly bilateral symmetry? Why is that? Which means like your right face is not exactly the mirror image of the left. This is because some cells on your on one side of your face divided probably one more times than the cells on the other side. So this is called the random effects. But but the influence from the random events are relatively smaller compared to the effects from the genetic effects and the environment. So we also need to consider the environmental effect, because sometimes the environmental effect may have a major may play a major role on the phenotypic variability. So, for example, if we look at the identical twins, so identical twins means they have exactly identical genetic makeup. If they grow up in the same environment, yeah, they will look very similar to each other, right? However, if the identical twins may behave quite differently, or if the twins are separated at birth and they grow up in a totally different environment, probably, yeah, these identical twins may look quite different here. So this example shows how the environmental effect may play an important role in the variability in the phenotypes. All right. So I don't think I want to spend too much time on this slide, because in genetics, if we want to study something, if we want to test some hypothesis, we need to do experiment. Then we're gonna leverage something called the model system, the small animals that we can use to test our hypothesis. For example, the small things, including the E. coli or yeast, the big things may be used just like the mouse, Drosophila, and rice. And the common features for this model system is they need to be small, because yeah, it's easy to handle in the lab, right? And the generation time should be short, and it should be easy for us to cross or mate those animals or plants, and they need to be they need to have a very small genome, because otherwise it's too difficult for us to study those organisms. All right. We still have 15 minutes. Let's take like 1 minute break. So any questions so far before we move on to the second topic? Okay. Okay, let's continue. All right. So before Mendel, there was a theorem called the blending theorem. What is the blending theorem? Because the people at that time, before Mendel, they came up with this idea to explain the inheritance patterns. The blending theorem means like the genetic materials just act like a fluid. For example, the two parents are crossed. So one has a red phenotype and the other one has a white phenotype. So all the progeny will have the trace in between these two phenotypes. Just like we mix two types of fluid together. But the problem is like, once you mix the two types of fluid together, you can never separate them again, right? So by using this theorem, so if you go through a very long time of the generation, so everyone will look like the same identical phenotypes. So this is predicted situation in a population. However, this is not the true in reality. So even after many generations and the generations, we still see a wide range of the variability in any of the traits. So that means this blending theorem does not make sense. So then Mendel proposed a theorem based on the particles. So what he proposed is that each individual has two particles. And when each individual produced the gametes, the two particles will separate, and only one particle will be passed to the progeny. One particles. And the same for the other parents. So the other parents has two particles carrying the genetic information. And when this parent produces the gametes, these two particles will separate, and only one particles will be passed to the next generation progeny. All right. And these two particles will form together to form each of the single progenies in the next generation. And now we knew these particles are called the alleles of the same gene. And the relationship between these two particles or two alleles, we can use the dominant or the recessive to describe the relationship between these two alleles. The dominant phenotype means this phenotype will be expressed in every single generations. So in every generations, you're gonna see these phenotypes. But for the recessive phenotypes, the reason why we call this recessive, because in some generations, these phenotypes may be masked. All right, so this is different theorem, particle theorem, compared to the fluid, the blending theorem. All right, so let's look at the experiments that Mendel did in his time. As I mentioned, he studied different phenotypes for the pea plants, the seed shape, the seed color, and the flower color. All right. And so, in fact, he actually studied more than 25 traits, but only these 7 traits showed the data that is in agreement with his hypothesis. So in his report, he only reported the data for the 7 traits. So now we understand why the rest of the traits did not show the data following his theorem, because those traits are complex traits, not like these 7 traits. These 7 traits are single traits because each trait is only controlled by a single gene. But the rest of the traits are called the complex trait because those traits are complex traits, which are controlled by more than one genes. For example, just like the human height. So this is not a single gene stuff. It's a complex trait that is controlled by more than one gene. Okay, so let's see how Mendel did his experiment. So the first experiment is called the selfing, which means the fertilization is done within a single plant. So what we can do is like we just transfer the pollen to the stigma within the same individual plant. All right, so then we call this inbred. Another experiment is called the cross-pollination. So, which means like the fertilization is done between 2 different plants. So first, we have to remove anthers from one flower to avoid the selfing, right? To avoid the fertilization. And then we transfer the pollen from another flower to this plant to complete the cross-pollination. And there's another definition is important called the true breeding line. The true breeding line means if you cross this line, you will always see one phenotype. You will not see the phenotype segregation, no change in the phenotype. So we call this is the true breeding lines. Otherwise, it is not. All right, so I think, yeah, time is almost up. And this is my first time to teach this lecture in the Zoom. So I think, yeah, maybe I could not finish all the slides today. I'm a little bit behind the schedule. I don't want to rush because, yeah, that will affect the quality of the lecture. So I'm gonna stop here so far. So if you have any questions, we can discuss now.
Hello, Professor.
Yeah, please. Yeah, go ahead.
Just really quick, can you go over homologous and heterologous chromosomes again, because I didn't quite pick up on that bit of the lecture.
Sure, no problem. Let me go back to here. Okay, so so to answer your question, let's use human as an example. So this is the chromosomes, all the chromosomes in a human cell, right? All right. So when we talk about this pair, these two chromosomes are called the homologous chromosomes because they are similar to each other and they carry the same genes at the same locus. So this is the homologous chromosomes.
Okay.
All right. Now, if you look at this one and this one, so what's the difference?
Well, I mean, for one, they're different sizes, right?
Yes, they're not different in size, but they're not only different in size, but also different in the genes, gene makeups. So these two chromosomes are non-homologous chromosomes because they carry totally different genes, right? Do you agree?
Yes.
Okay. So so I hope I answered the question. So this is this is how you tell the difference between the homologous chromosomes and the non-homologous chromosomes.
Okay, and then over here to the left, where you have where we have our genes where they're the same shape but different colors. Would that make them homologous or heterologous?
Oh, okay. So so I think, yeah. So we were using two different terms here. So when we talk about the homologous or non-homologous chromosomes, we are only talking about the chromosome levels, just like this. So those two, because they are in the same pair, right? So they are homologous chromosomes because these two chromosomes are very similar to each other. They are similar in length and they're similar in the gene makeups on these two chromosomes. However, if you compare the chromosomes in different pairs, they are non-homologous chromosomes. Yeah, because they're different, right? But when we look into a chromosome pair, so these two are homologous chromosomes, right?
Yes.
But when we look into, we zoom in the homologous chromosomes, and when we look at just a single locus on these two homologous chromosomes, then we use different terms to describe the genes at this locus. So we use homozygous, Zygous.
Okay.
Zygous. Oops. It's hard to write like this. So okay. So now we use homozygous or heterozygous. These are two different terms, not like the homologous chromosomes. Homozygous or heterozygous to describe the feature of a specific loci on the homologous chromosomes. Did I make it clear?
Yeah, so what you're saying is that that when we're talking about homozygous, we're talking about a specific pair. We're not talking in between pairs. And in this pair, we're talking about whether or not a locus is different or the same as the locus from the other chromosome in its pair.
Yes.
Okay. All right. And in this case, the blue squares are homozygous, but the yellow and pink are heterozygous, and the blue and pink are also heterozygous.
That is true. Yeah.
But at the same time, they are homologous.
Yes. Yes.
Okay. Okay.
So
Gotcha.
we only talk about the heterozygous or homozygous loci within the homologous chromosomes. Yeah, if two chromosomes are not homologous at all, so they even do not carry the same gene, so we cannot talk about the heterozygous or homozygous loci, right?
Yes.
Yeah, so yeah, so that's the basis. So we only look at the homologous chromosomes to check each loci to see if these two homologous chromosomes carry the same alleles or different alleles at each locus.
Sure.
You're right. So the example showing here, the square, is showing the same color, which means at this locus of these two homologous chromosomes, this gene has the identical alleles. So we call this situation as the homozygous locus, homozygous locus. But if you look at the other two, the circle and the triangle, they are showing different color. Means for these two genes, the two homologous chromosomes have different alleles. Then we use heterozygous to describe these two loci.
Okay. Yeah, I think I get it now. Thank you so much.
Great. Okay. You're welcome. So any other questions?
Just to repeat his last question, just to make sure. So homologous has to do with gene, and the homozygous has to do with the allele.
Yes.
Okay.
Yes. Very good.
All right. Thank you, Professor. You have a wonderful day. I'll see you in 2 days.
Thank you. You too. Thank
Bye-bye.
you. So I think, yeah, let's see, time is up, right? So yeah, today, I think, yeah, I'm gonna end the lecture today now. Sorry, so this is my first time, so I think, yeah, maybe next time I need to, I try to find a trade-off, like, I don't want to talk too fast, because sometimes, yeah, we'll get you lost, but sometimes, like, but at the same time, we have many topics to cover. So today, this left me a little bit behind my schedule. So I'm going to think about a better way to catch up next time. Thank you so much, everyone. See you on Thursday.
Professor,
Yes.
I have a question about the uploading of the lecture.
Yes.
Like,
Yeah.
could you upload the lectures on the day of the lecture, like the recorded one?
Could you say that again? Sorry, I cannot hear you very clearly.
Oh, sorry. Can you upload the recording of the lecture on the day of the lecture? Because sometimes, like, I'm busy, right, and won't be able to make the lecture. And having like schedule stay as normal instead of uploading it on the weekend would help me stay on track.
Sorry, I still cannot hear you. So you are talking about the schedule for uploading the recorded lecture, right?
Yeah.
Yeah. So what is your suggestion?
Can you upload it on the day of the lecture, the recording?
Oh, you mean on the same day,
Yeah,
during the night? Yeah, I will try to do that. Sure,
Okay, all right, yeah, thank you.
sure. Okay.